Hello everyone. Today we will talk about oxidative phosphorylation. So last time we talked about electron transport, and now it is time to talk about how this is connected to the production of ATP. So people who were trying to figure out the electron transport chain did so by doing experiments where they had cells in culture, and then they would put in different inhibitors to see if they could inhibit the transport chain, and then if you could add other substances to see if you could rescue the electron transport chain. So I have a number of inhibitors here. So Rotenone and amytal are small organic molecules that inhibit the electron flow between complex 1 and coenzyme Q. Antimycin A blocks electron flow between the reduced coenzyme Q and cytochrome C1. And cyanide, azide, and carbon monoxide all block electron flow from complex 4 to oxygen. So here are the structures of all of those compounds. They don't seem to have anything in common, but they do inhibit the electron transport chain at the positions that I indicated in the last slide. So the way that we can visualize this happening is to use this water pipes analogy. So I have a scan from an old textbook here. So imagine the electron transport chain is analogous to some pipes with water. So you have a tap up here where water is flowing, and then we have an open tube right here where the water flows out. So water flows in here and out down here. So water flows freely as long as the pipes aren't blocked. Now suppose we added rotenone and amytal. So if we, again, imagine that the electron flow between complex 1, so this is the flavoprotein involved in complex 1, and coenzyme Q is blocked, you can see that the tap continues to have water flowing here, but these pipes will fill up. So you could, though, bypass this. If you took your tap and you moved it, if you moved it over here to this pipe right here, then you can bypass the block and then water can continue to flow out of the pipe. So biochemically, if you had added rotenone or amytal to your culture, you would see that electron transport was blocked. But then if you added succinate, then succinate could donate electrons to complex 2, which is below the block, and then complex 2 would donate its electrons to coenzyme Q, and then you would get around the block that way. So here's another way of visualizing it, that if we have this block right here of rotenone or amytal, we block it right here, but succinate would give its electrons to FAD and then coenzyme Q, and then you'd be able to continue the electron transport chain in the place it's highlighted in pink. So what if we added antimycin A? Again, if we did that, that involves blocking the electrons between QH2 and cytochrome C. And again, you can see that the tap would fill up the pipes ahead. But again, you could bypass this. You could take this tap and you could move it over here and have it flow into this cytochrome C pipe. Biochemically, what that means is that if you added antimycin A to your culture, you could that would inhibit electron transport, but then you could rescue it by adding an artificial substrate, and that's ascorbate or vitamin C, and vitamin C can donate electrons to cytochrome C. So again, you bypass the block, and then if you have vitamin C right here and it's adding electrons to cytochrome C, then water can continue to flow. So again, if we're visualizing this here, we have our antimycin A block right here between cytochrome B, which is part of complex 3, and cytochrome C1. It's not marked that way here, but that's what it is. It's cytochrome C1 rather than cytochrome C. Then you can bypass the block by having vitamin C add electrons to cytochrome C, and then you would be able to continue electron transport to the final electron acceptor, oxygen. The problem becomes with these substances, carbon monoxide, azide, and cyanide, they block transfer right at the end. So if I have my little diagram right here, that's the last step in complex 4. where the cytochromes A and A3 are donating electrons, and finally to oxygen to form water. So here, there really isn't a way to bypass the block. You don't have any more pipes left. This explains in part why carbon monoxide, azide, and cyanide are such deadly poisons, because how they act on the electron transport chain. They have other effects too, biochemically, but this is one explanation for why these are so deadly. So once the investigators figured out how the electron transport chain worked, then the linkage between electron transport and oxidative phosphorylation was a bit tricky to figure out. So in electron transport, we have all of our electrons and protons from our metabolic fuel. So we focused on glucose, but we also saw that lipids can break down to acetyl-CoA, and proteins can break down amino acids, and they can feed into pyruvate and acetyl-CoA as well. So we have all of our coenzymes, and they have donated their protons and electrons all the way to the electron transport chain to oxygen to form water. So that's the electron transport piece of this. But we did the calculation where we saw that that was an exothermic process, or sorry, I should say exorganic process, sorry. So the free energy released in this process will be converted into a useful form. We will capture some of that energy and use that to synthesize ATP. And the term we use there, remember, is oxidative phosphorylation. So this is the term that we use to describe how ATP is synthesized. So there were lots of hypotheses put forth to describe how this worked, but Mitchell's chemiosmotic theory of how electron transport is coupled with oxidative phosphorylation is consistent with all the data that you can collect on this. So what happens is, we did say last time when I was telling you about all these complexes that are in the inner mitochondrial membrane, I mentioned to you that complexes 1, 3, and 4 participated in proton pumping. So if you're pumping protons from the matrix into the inner membrane space, what happens is that establishes an electrochemical gradient across the intermembrane. So protons are positive. So if you pump a positively charged entity out of the matrix and into the intermembrane space, then the intermembrane space becomes more positive relative to the intermembrane space. So we have a change there, but we also have a change in pH. So if we have a high concentration of protons in the intermembrane space, that will have a lower pH than in the matrix. So the matrix will have a low concentration of H+ and a higher pH. The chemiosmotic theory states that the electron flow, and in this figure, electron flow is represented by this blue line, and here the emphasis is on NADH. So we saw NADH passes electrons through complex 1 to coenzyme Q, to complex 3 to cytochrome C, to complex 4, and then finally oxygen to form water. So that path is shown in blue. The FAD, remember, gives its protons and electrons, succinate actually gives its protons and electrons to fumarate, and in so doing, FAD accepts them to form FADH2 in complex 2, and then those electrons are transported to Q. And then the rest of the pathway is the same. So the blue lines show electron flow. What is shown in pink is the proton pumping. So complex 1 pumps 4 protons, complex 3 pumps 4 protons, and complex 4 pumps 2 protons, for a total of 10 protons for every 1 NADH. Notice that complex 2 does not participate in proton pumping. So again, what we have is we have this electrochemical gradient with a more positive charge on the intermembrane space side, and a more negative charge on the matrix side. What will happen is these protons will, because this membrane is impermeable to them, these protons will flow through. This is the ATP synthase enzyme right here. So this is called F0, so that's the channel by which these protons can flow. And this is F1, and that's the catalytic unit. So when the protons flow through the F sub 0, they activate F sub 1, and the activation of this enzyme allows ADP and phosphate to be coupled to form ATP. So the electron flow is accompanied by the pumping of these protons, which forms this electrochemical gradient, and that activates the active site of ATP synthase. So that allows ATP synthesis to occur in the presence of proton pumping, which occurs during electron transport. So this is a picture from our book. So again, to orient ourselves, here the matrix is on top. So the protons are being pumped through complex 1, complex 3, and complex 4. So we have an excess of protons, positive charges in the inner membrane space, and a deficit of protons in the matrix. So this is referred to as the proton motive force. This potential difference across the membrane is an energy source that is tapped by the ATP synthase in order to activate it to be able to synthesize ATP. So this gradient is both electrical and chemical. This imbalance of protons is a non-equilibrium state. So this proton motive force has an associated free energy with it. And the associated free energy is the energy that it would cost us to restore the system to equilibrium. So we can calculate that free energy. So first we would have to look at the chemical part of the electrochemical gradient. So we could measure the concentrations of the protons that are pumped out and express it as this ratio to the concentration of the protons that are in the matrix. We should also address the chemical portion of the electrochemical gradient. So here, delta G is Z, this is the Faraday, and delta psi. So Z is the charge on the proton, and delta psi is the membrane potential. So you can actually go into the laboratory, and if you have respiring mitochondria in culture, the membrane potential is something that you can directly measure. So that means then this free energy associated with the proton motive force, or this electrochemical gradient, this imbalance of protons across the membrane, the associated free energy is expressed by this equation. So this piece is the chemical difference, and this piece is the electrical difference. So here's another picture right here. So again, to get ourselves oriented, protons are being pumped out of the matrix. So over here, this is the matrix, out of the matrix and into the inner membrane space. So we have a higher concentration of protons, and this author calls this the P side, or the inner membrane space. And over here, this is the matrix. So we have a deficit of protons over here. So that means we have a lower pH here and a higher pH here. And again, the authors here show how we can calculate the free energy associated with the potential difference across that membrane. Well, what we should notice is passage of 10 protons. So again, that's our 10. So that's 4 protons in complex 1, 4 protons in complex 3, 2 protons in complex 4, that's our 10 protons, and that generates a proton motive force of about 200 kilojoules per mole. So that is the energy captured by the exergonic transfer of the NADH's, protons and electrons, all the way down to oxygen to form water. So this proton motive force then serves as the energy reservoir which drives ATP synthesis. So we can calculate this. So we have done this before, if you remember, that if we took the NADH and looked at its ability to donate protons and electrons to oxygen to form water, remember in a previous exercise we calculated the E value of that entire reaction from the reduction half potentials, and we got a delta G 0 primed of negative 220 kilojoules per mole. So you can see that is similar in magnitude to the delta G that we can calculate by the establishment of that electrochemical gradient. So this is what we've done. We've taken the free energy of this reaction and we've captured it in the form of an electrochemical gradient across the membrane. So we haven't just completely dissipated the energy, we've captured it in the form of that potential difference across the membrane. So I'm going to modify this in a bit, but for right now, it's maybe a little bit comforting to work with integers, whole numbers. So complexes 1, 3, and 4 participate in the pumping, and the numbers aren't shown here, but it's 4 protons for complex 1, 4 protons for complex 3, 2 protons for complex 4, for a total of 10 protons for every NADH that participates in electron transport and donates its protons and electrons to oxygen to form water. And again, those protons are going to come across this F sub 0 channel and stimulate the ATP synthase. So the way we can look at this for now is that we get about 1 ATP per complex that participates in proton pumping. So that means for NADH, there are 3 complexes that participate in the pumping, so we get 3 ATPs for every NADH. So we can look at that here too. So if we look at the free energy drops, so here's complex 1 shown in blue, here's complex 3 shown in orange, and here's complex 4 shown in green. And if we look at the energy drops right here, the redox potentials are shown over here, and converting those to delta G. Now this is kilocalories because it's an old figure, not kilojoules. But what we can notice, and maybe that's diagrammed better here because this is kilojoules, is complex 1, complex 3, and complex 4. If you look at these energy drops right here that occur when the electrons are transferred down the electron transport chain, each of these releases sufficient energy to synthesize an ATP. So if it only costs us 30.5 kilojoules per mole to make an ATP, we have enough energy in each one of these complexes in order to synthesize 1 ATP and a little left over. Okay, so we want to couple this to the activation of ATP synthase. So I'm going to go to this figure. You do have to enable the Adobe Flash Player. Okay, so there's our mitochondrion. So remember, electron transport occurs in the inner mitochondrial membrane. So here's the inner mitochondrial membrane, here's the inner membrane space, there's the outer membrane, there's the cytosol. And we have our complexes. Now here the author refers to this as complex 5. I prefer to call it ATP synthase because that describes what it does. But here this green arrow shows the electrons and protons being passed down the electron transport chain to the final electron acceptor, oxygen, to form water. And in so doing, 4, 4, and 2 protons are pumped through complexes 1, 3, and 4 respectively. And again, now we have a buildup of protons, positive charge in the inner membrane space, and a deficit in the matrix. So what happens then is these protons are directed toward into this F sub 0, which is not shown here, but that's the channel across the membrane, and then the active site is located in the matrix, and when that's activated, ADP and phosphate are coupled together to make ATP. So this simple animation shows us how electron transport, shown here in this blue line, is coupled to the synthesis of ATP. And this is a little bit nicer of a diagram that shows protons being, they flow through the ATP synthase to activate it and synthesize ATP. So I have a number of pictures here, but the best way to look at this is in a video that I can't show on my screencast program, and because I did try it and it produces this very echoey sound, so you'll have to do this on your own. This is from our book, and this shows a schematic diagram of what the ATP synthase looks like. So again, here's the matrix. Protons are pumped out of the matrix to the inner membrane space. And there's the F sub 0 channel right here. And there is a shaft right here that is connected to the F sub 1. And there are 6 subunits right here. There are 3 alphas and 3 betas. And you can see this under the electron microscope. It looks kind of like lollipops, where you can see this sort of this stalk right here and these little sort of circular things right here. So again, to blow that up, there's a gamma subunit right here that's attached to these 3 alpha and 3 beta subunits. And we do have some idea of what they look like. This is the X-ray crystallographic structure right here that shows this is the channel. So lots of alpha helices here. And here's that gamma subunit shown in purple. And then the alpha and beta subunits are shown. I think beta is in green and alpha is in blue. I'm not sure about that, but they're each shown in a different color. So we have 3 alpha and 3 beta subunits. So the active site is mainly located on the beta subunit, and there are 3 of them. And the 3 of those subunits can exist in 3 different conformations: loose, tight, and open. In the loose form, ADP and phosphate are bound. In the tight configuration, the reaction has occurred to couple ADP and phosphate to make ATP. And in the open portion right here, the ATP is released and now new ADP and phosphate can bind. So we have 3 beta subunits, 3 different conformations, and you never have more than 1 at a time. During electron transport, these conformations change in this order from loose to tight to open and then back to loose. I have an animated figure that I wish went through an entire cycle, but unfortunately it doesn't. So here we're going to start with, there are 3 beta subunits. So we have loose, tight, open, and then this is the gamma subunit right there that's going to turn them around. So when we start here, the tight subunit has ATP bound, so it has just finished synthesizing an ATP. So ADP and phosphate can bind to the loose configuration. So keep track of what's happening because this will show in the video. So ADP and phosphate are bound to the loose configuration. ATP is bound to the tight configuration. So what happens next is we have the energy of electron transport. And when that happened, the protons that were pumped out of the matrix and into the intermembrane space came through that F sub 0 channel and then rotated this. So now the conformations changed. So it was loose to tight to open. So this was loose and now it's tight. And this was open. This was tight, so now it's open. And then this was open, so now it's loose. So what can happen now is that now that this is open, the ATP can leave. And now that this is tight, the ADP and phosphate can form ATP. So the next frame will show that. So that ATP leaves, and we have a new ATP being synthesized. So that is the end of the animation. I do wish they would have shown an entire rotation. So we didn't get to see an entire progression. An entire cycle of this. But what happens, which is shown in this diagram here, is that we have 3 subunits, 3 beta subunits. They go through an entire rotation. So that means each subunit in turn progresses from loose to tight to open. Each subunit makes a single ATP. So what that means then is if this undergoes 1 complete rotation, and we have 3 subunits, and every subunit makes an ATP, then we can make 3 molecules of ATP per 1 rotation. So I just want to remind you that I am using whole numbers right now. I'm going to change that in a bit, but right now I think it's instructive to do it that way. So I wanted to show you the mechanism right here. So this is how the ATP synthase works. So the alpha subunit is shown in green, so you can see that largely the beta subunit is where the active site is, but of course the alpha subunit is close by and plays a role in catalysis. So we have two residues, so the ARG182 and ARG376 on the beta chain. You might remember that those are positively charged, and we are over here, we're making ATP from ADP and phosphate. So we have quite a lot of negative charges here. So that means that these ARG residues, so here's one ARG residue right here, and then there's and there are our lyse residues, which are also positive. So we have lysine, arginine, and another arginine over here. And what we can do is we can have those positive charges along with the magnesium, which is also positively charged. All of these things are able to stabilize the negative charge on the phosphate. So that's the phosphate that I'm circling right now. That's the phosphate, and it has two negative charges. And then here's the ADP, and the ADP has negative charges too. So this active site is doing what all enzymes do. It's forming these ion pairs in order to stabilize the transition state. Remember, we have a transition state stabilization. That's what enzymes do. So all of these positive charges are helping to stabilize the transition state. So I don't have a video of this, but what will happen is that the ADP and the phosphate will be coupled to form ATP. So ATP is a product, and water is a product too. So over here, they show the substrates, and then that's where the water would be before it left the enzyme, and the glutamate, which is negatively charged, is able to form a hydrogen bond with the water right before it leaves. So this provides a nice snapshot for how the ATP synthase works. So I have some videos. So there's a video that I'm just mousing over right here, and another video that I'm mousing over right here. So these videos are on Sakai, and again, I can't put them on my screencast program, unfortunately, but what you need to do is you need to stop this PowerPoint and go and watch them right now, because this shows beautifully how when the protons are pumped out of the matrix and into the intermembrane space, they flow through this channel right here, and this shows how when the protons bind, you get this rotation of this complex right here, and then that rotation is coupled to these subunits right here, and then this causes the rotation, the loose to tight to open rotation that I showed you on the last slide. So in green here, we have the 3 alphas and the 3 beta subunits. So the protons are binding right here, and that turns this complex, which then is connected to this protein piece right here. This gamma subunit protein then rotates, and then this whole thing rotates. So if we get a full rotation, then all 3 of the betas can be loose and tight and open, and each beta can synthesize 1 ATP. So all 3 beta subunits change conformation during electron transport. So that means that in order to synthesize ATP, we have to have electron transport. We have to have a proton gradient, because the proton gradient, the protons coming back through that F sub 0 channel, that's required to drive the rotation of that gamma subunit, and that's what causes the rotation of the F sub 1, those 3 alpha and 3 beta subunits. So let's define something called the P2o ratio. So that describes the relationship between respiration and ATP synthesis. So that's going to be defined as the amount of ATP synthesized per atom of oxygen reduced. So the way I've been presenting it so far, that means that if I have a half an O2, so a half an O2, that's 1 atom of oxygen. And if I get 2 protons and 2 electrons from 1 NADH when that happens, and I get 1 whole rotation when that happens, then I get 3 ATPs. So the P2O ratio for NADH would be 3. So again, to connect that to electron transport, so when NADH goes through electron transport, it goes through complexes 1, 3, and 4. In total, 10 protons are translocated into the intermembrane space. one complete rotation of gamma occurs, all three of the ATP synthesizing units, that means the beta subunits of the F sub 1 synthesize an ATP. So the P to O ratio is about 3. So again, I'm going to change that. Don't get attached to this, but right now it's instructive to go through these things as whole numbers, I think. So what about FADH2 and that succinate to fumarate reaction? Well, remember we were going from succinate to fumarate and then through complex 2, which doesn't pump any protons. So you go through succinate to fumarate to FADH2 to QH2, and then through complex 3 and complex 4. So that way you only get 6 protons. So that only gives you a 2/3 rotation. So only 2 of the beta subunits make an ATP. So that gives you 2 ATPs. So here the P to O ratio is about 2. All right, so here I'm going to change those numbers from the nice integers. It's very nice to think of 3 complexes and 3 beta subunits and 3 ATPs. But under physiological conditions, there's a little bit of proton leakage. So that means the P to O ratio isn't really 3. It's about 2.5. And for QH2, it's not 2, it's about 1.5. So I wanted you to be aware of this. I still see some, even though most of the textbooks should have updated this to, this is not brand new information, but sometimes I still see P to O ratios of 3 or for NADH and P to O ratios of 2 for QH2. But hopefully in your future, you'll see that this has been updated. So one of the things I asked you to do in your top hat exercise for this morning is I wanted you to go through and count how many ATPs you get per 1 mole of glucose. And the way we do this is we look at both oxidative phosphorylation and substrate level phosphorylation. And we use these P to O ratios. So glucose to pyruvate gives us 2 NADH and 2 ATPs, so that's substrate level phosphorylation. Pyruvate to acetyl-CoA gives us 2 NADHs, and then 2 acetyl-CoAs go around twice, and we get 6 NADHs, 2 FADH2s, and 2 GTPs. So if we do the math, this is what we get. Remember that this NADH right here had to be transported into the mitochondrial matrix from the cytosol. So if we use that malate aspartate shuttle, we didn't lose any energy. So that means that we get a total of 32. So we get, again, so 2 times 2.5 is 5, plus 2. 2 times 2.5 is 5 here. 6 times 2.5 is 15. 2 times 1.5 is 3, and then this is substrate level phosphorylation. Add it all up and you get 32 ATPs. So notice that you get 28 ATPs from oxidative phosphorylation and 4 from substrate level phosphorylation. So having oxygen gives you a big advantage in getting the most out of your glucose. Remember that we also talked about the glycerol phosphate shuttle, and there the NADH donated its electrons eventually to FADH2. So there was a proton loss there. So here, instead of considering protons from NADH, we'd have to do FADH2. So we'd have to take this 2 and multiply by 1.5. So this would be a 3, but everything else would be the same. So here, if we use the glycerol phosphate shuttle, we get 30 ATPs. So what else do we have to think about? Well, we have to think about these other transporters because we need to, okay, here's our ATP synthase. There's the proton gradient that allows this F sub 1 to be activated to have phosphate and ADP be synthesized to ATP. So that means we need phosphate as a reactant and ADP as a reactant. So we have this phosphate translocase. So that brings in phosphate and a proton. And then the phosphate then serves as the reactant for the ATP synthase. Here we have an antiport where ADP comes in and ATP goes out. So the ADP is serving as a reactant. So we have phosphate and ADP as our reactant, and then that's our product. So the product leaves because ATP will be needed in the cytosol. So how do we know that this chemiosmotic hypothesis is correct? Well, we have some evidence. So one is that you can measure the pH of the intermembrane space, and you can find that it's lower because it has all those protons. I don't exactly know how you would do this, but you can impose a pH gradient, and if you do that, you can get ATP synthase, but you'd have to artificially impose this experimentally in some way. The other thing is that you need a closed compartment. So if you throw in detergent and disrupt your membranes, this is not going to work. And the last thing is there are uncouplers that dissipate the gradient. So the most classically examined uncoupler is DNP. So DNP is dinitrophenol, and this dissipates the proton gradient and it uncouples electron transport from oxidative phosphorylation. This means that electron transport still occurs, but you don't get any ATP. And we know, because we calculated this, that electron transport has an associated free energy with it, and that it is an exergonic process. So that means if you're not capturing that energy to synthesize ATP, then you're losing it as heat instead. So you would not think that dinitrophenol, maybe you wouldn't think it's very lipid soluble, but it is. But you might even think that if you lost a proton to form the dinitrophenolate, you look at that charge there, and you wouldn't think that would be membrane soluble either. But they both are. And the way that we can explain this is that the dinitrophenolate anion, we can draw 3 resonance structures for this. And what you see is that these resonance structures all have charges, formal charges associated with them, but the charge is delocalized. So that means that dinitrophenol can pass through a membrane very easily. It just literally dissolves in the membrane. And then dinitrophenolate can too. So this is how it works. So if I am, again, to get us oriented here, there's our inner mitochondrial membrane, and the electron transport happens, and the protons are supposed to be pumped out of the matrix and into the out of the matrix and into the intermembrane space, and then those protons go through ATP synthase to stimulate the F sub 1. So those are all those beta subunits right there, and they synthesize ATP. But this is what happens if you have, if you add DNP to your culture. So here's DNP, and DNP is protonated, but it can cross the membrane because it's lipid soluble and go into the matrix. And in the matrix, it's going to encounter an environment where there's a lower proton concentration. So that's going to shift the equilibrium to having this dinitrophenol release a proton. So now we have the dinitrophenolate anion, and then what it can do is, because the charge is delocalized, it can go back, and this dinitrophenolate anion then will encounter the intermembrane space that has a high proton concentration, and it becomes protonated. So what happens is the dinitrophenol, essentially, the protons are being pumped out of the matrix into the intermembrane space, but what happens is the dinitrophenol just carries them right back in. And if the dinitrophenol is going through the cycle and dragging the protons back in, then the protons aren't going to go through the ATP synthase. And the protons don't go through the ATP synthase, you get no ATP. So again, it's important to remember, though, even though you wouldn't think this could cross the membrane, it can, because its charge, its formal charge, is delocalized via resonance. So I want to leave you with one last thing, and that is how we control oxidative phosphorylation. So hydrolysis of ATP to ADP in a resting individual is low, but if you get up and do some activity, then hydrolysis of ATP increases. And that increases the ADP concentration, which increases the rate of electron transport. So we can go into the lab and we can do this experiment where we measure electron transport rate by measuring oxygen consumption. This is a really good way to measure the rate of electron transport, because oxygen is the final electron acceptor. So the more oxygen you consume, the greater the rate of electron transport. So this slope then over time tells us the rate of electron transport. And then if we add ADP, you notice there's this huge change in the slope and a huge increase in oxygen consumed over time. And then as all that ADP is converted to ATP, then it levels off again. So again, measuring oxygen, the fine electron acceptor, is a good way to measure the rate of electron transport. And electron transport is rapid when the concentration of ADP is high, and it slows down when ADP drops. So this is going to cause electron transport to be very, very rapid if you had DNP. Because if you have DNP, remember you have the DNP shuttling the protons, and you're not getting ATP synthase. So that means ADP concentration is going to go way up. So that's the other thing that happens when you add DNP, is that you have a very rapid rate of electron transport. So that means all of this is coordinated. So glycolysis, the citric acid cycle, electron transport are all coordinated. So the citric acid cycle and glycolysis produce high concentration of NADH and FADH2. So electron transport will be stimulated by the high concentrations of those reduced coenzymes because they're feeding into electron transport. We'll find out in the next chapter that fatty acids, when they break down, produce acetyl-CoA. So acetyl-CoA goes into the citric acid cycle, and you get more NADH and FADH2 there. So lots and lots of NADH and FADH2 will stimulate electron transport. So so you've probably seen BioVision's Powering the Cell, because we all love that video. So I will let you look at that on your own. So you should look at that now, even if you've seen it before, because you might not have recognized ATP synthase, but it's in there, and you'll recognize it now. So what we can do is we can look at how we put all of this together. So in glycolysis, remember we said that hexokinase was regulated, and then glucose 6-phosphate acted as a feedback inhibitor. And then the phosphofructokinase is very highly regulated by its reactants, phosphate, AMP, ammonium ions. That's going to be tied in with amino acid metabolism, which we haven't gotten to yet. Inhibited by ATP, inhibited by citrate. So ATP and citrate means that energy resources are plentiful, and you don't need to have this on so high. Fructose 1,6-bisphosphate will stimulate. All right, then we got to pyruvate kinase. So pyruvate kinase is stimulated by ADP and inhibited by ATP. Again, other indications of cellular energy. And then we get to pyruvate being converted to acetyl-CoA. So we talked about the pyruvate dehydrogenase complex and how it is highly regulated by pyruvate. The products stimulate, so ADP, calcium, they indicate that energy is required, whereas acetyl-CoA and a high concentration of NADH means the energy is not required. So those are inhibitors. And then we go to the citric acid cycle, and we talked about how those enzymes are regulated. So we have the citrate synthase, the isocitrate dehydrogenase, and the alpha-ketoglutarate dehydrogenase enzymes. Again, those are the 3 irreversible reactions in the citric acid cycle, and we talked about that in chapter 14. And that is the end. So again, that's something else that I wish that they had continued, because once you get through glycolysis, the pyruvate dehydrogenase reaction, and the citric acid cycle, if you see here that you have really high concentrations of NADH and FADH2, those will feed into electron transport to produce ATP. So again, you'll have high electron transport rate if you have lots of those coenzymes, but things slow down if you generate sufficient ATP to power the organism. So again, I want to point you to some more videos. This is a guided exploration that provides a nice overview of electron transport. This is a video that describes the Q cycle. So I would like to encourage you to watch that as well. So this is a really nice, I think, figure. So I'll let you study this on your own. That shows how it doesn't show a glycolysis, but it shows pyruvate entering through its transporter and then going through the citric acid cycle. And here are those transporters I showed you. Here's the electron transport chain highlighted in blue. There's our ATP synthase right here. And all of these are shown by these colors here. So I will let you look at that. But that's a really nice summary, I think, of both chapters 14 and 15. So I hope that you have enjoyed chapter 15. I am very sorry that we will not be able to play the Electron Transport game. It's usually a lot of fun, and I'll miss not playing it with you. But any of you who are juniors, you can come back in the fall and join my class, and I'll be happy to have you. So I hope you enjoyed this, and I will see you next time.